All right, so this video is on lipids, which is one of the major macromolecules, and they're also known as fats, oils, and the signaling molecules, steroids. And the way you can tell all liquids is they share the common trait of being hydrophobic. Think back to what we talked about when we talked about the properties of water. What does it mean to be hydrophobic? Remember, things that are polar tend to dissolve in water. Things that are nonpolar tend to not dissolve in water, and those things are hydrophobic. And we'll talk a little bit about why that is in a second. First thing, we have to know the rules for identifying a lipid. First, it's a carbon to hydrogen and oxygen, so it's made of 3 elements mostly, in a 1 to 2 to very, very little oxygen ratio. So for every 1 carbon, there's 2 hydrogen, but in terms of the number of oxygen, it's really low compared to the other 2. Because of that, you have long chains of carbon and hydrogen, which are called hydrocarbons. And if you remember back when we talked about carbon chains, hydrocarbons are very hydrophobic. It's not until you start adding other things to it that they become more hydrophilic or polar. Another characteristic is sometimes they contain phosphorus and nitrogen, but that's in a special case called phospholipids, and you can still tell in phospholipids that it's a lipid because of the long chains of carbon and hydrogen that they have, even though they also have nitrogen and phosphorus. So let's take a quick look at a triglyceride. So this is a triglyceride, which is your standard fat that floats around in your blood. It's called a triglyceride because it has a glycerol molecule right here, and it's connected to 3 fatty acids. So that's where the tri part comes from. Notice when you put it together, you do a dehydration synthesis reaction. You put it together, water comes out. Now let's look at the rules that we talked about before to show that this is, in fact, a lipid molecule. First thing, long chains of carbon and hydrogen. You can see them right here, long chains of just carbons and hydrogen. Notice the ratio. For every carbon, there's 2 hydrogens. But look here, we have only 1, 2, 3, 4 oxygen molecules. Here we have 1, 2, 3, 4, 5, 6 oxygen molecules. So way more carbons and hydrogens in comparison to the oxygen. Now when we classify these fatty acids, we classify them in 2 main ways. Are they saturated or are they unsaturated? And you may have heard of this before. So saturated fats have the maximum number of hydrogens connected to the carbons. So what that means is that there's all single bonds between the carbons here. And that makes it a very linear molecule, as you can see here. And when they're linear, it allows for it to stack up. And when they stack up, they do not flow as well across each other. So they're not as liquid. They're not as fluid. So these tend to be more solid at higher temperatures than, say, unsaturated fatty acids, which we're going to look at in a second. So these, when you think about saturated fats, they tend to be bad for you, right? That's because they have a higher likelihood, because they are more solid at room temperature, to collect in your arteries. Unsaturated fats, though, are the good fats you may have heard of. They have at least 1 double bond, you can see here. And when you have a double bond, that tends to kink the fatty acid. And when it's kinked, it does not stack as well. So this would not stack on top of another one as neatly. And as a result, it flows better, so it's actually a liquid at lower temperatures than saturated fats would be. Polyunsaturated fats just have multiple double bonds, and they stack even more poorly, and as a result, they flow even better. Okay, so remember, double bonds is what causes it to be unsaturated. If it's all single bonds, it's a saturated fat. All right, here's that special case I was mentioning, phospholipids. Now, the first thing you look for, long chains of carbon and hydrogen. What do you see down here? Long chains of carbon and hydrogen. This part is very nonpolar, so it's hydrophobic. They're called the hydrophobic tails of a phospholipid. The top part is called the hydrophilic head. Now, why is it hydrophilic? Notice it's charged, so that draws electron density to different places within the molecule, making it polar. But also you have lots of electronegative oxygens here to draw electronegativity towards it. Therefore, this part is more polar. This part's nonpolar. You may remember back when Ms. Bebeck taught this freshman year, or whoever taught you biology, that this is a component of the cell membrane. And the cell membrane's for this lipid bilayer. But why do they orient this way? It's because they have these hydrophilic heads and these hydrophobic tails. The hydrophilic heads will always orient themselves towards water because they're hydrophilic, they love water, and the hydrophobic tails will orient towards themselves. Another word for being both hydrophobic and hydrophilic, or having polar parts and nonpolar parts to a molecule, is called being amphipathic. You kind of want to remember that word. It's a really good word to kind of throw out there on the AP Bio exam if you're ever talking about a cell membrane or phospholipids specifically. Okay? Steroids are kind of a special case. So steroids are still long chains of carbon and hydrogen, similar to the other things that we've been looking at, but they tend to form ring structures instead of being linear. If you ever see a stick diagram like this, okay, every time you see a corner, that's a carbon attached to hydrogen atoms. So really, this is all just carbons and hydrogens throughout this whole entire molecule, with the exception of right here. Lots of carbons and hydrogens, very little oxygen, so 1 to 2 to very, very little oxygen ratio. Now, this is actually looking at cholesterol. So cholesterol is a steroid, but it is also the precursor for some hormones. So cholesterol actually gets turned into things like testosterone, estrogen, stuff like that. So remember, these are special because they don't look like other lipids, but it's still long chains of carbon and hydrogen. Remember, every time you see a corner, that's just carbons and hydrogens. If it's something other than carbon and hydrogen, they'll note it as being an oxygen or a nitrogen or phosphorus or something like that. Okay? Here's showing you testosterone. Here's showing you progesterone. There's estrogen. Those are all examples of steroids. So quick review of this stuff. Carbon, hydrogen, oxygen in a 1 to 2 to very little oxygen ratio. Long chains of carbon and hydrogen, making at least part of the molecule very nonpolar and hydrophobic. There's 3 types that we talked about: triglycerides, glycerol and 3 fatty acid molecules, phospholipids, which look similar to a triglyceride, but it has that hydrophilic head part that is charged and has other electronegative elements on it. And then steroids, which are those connected rings that have long chains of carbon and hydrogen that are used as signaling molecules in our body. Remember also, fatty acids can be saturated. Either they have all single bonds or they are unsaturated, where they have 1 or more double bonds between it. Remember, saturated fats tend to be solid at room temperature because they stack nicely because they're so linear. Unsaturated fats tend to be liquid at room temperature because of their bent nature. They do not stack as well on top of each other and thus flow. Okay, that's all for lipids.